Okay, thank you very much, Mr. Chairman. So, leading on from Jeff's wonderful talk around immune modulating therapies, I'm going to talk about other therapies that are not focused on the immune system. As I said earlier, I do have two academic conflicts, and these are relevant because I will be talking about mesenchymal stromal cells. We don't have a therapy. I think Dr. Bellingham showed us that very clearly. We have tried lots of things and they still don't work, and the original description by Ashbaugh and Petty is still true. We sort of fool around with ventilators and fluids and try drugs, but we don't seem to have a lot of success with the drugs part. This is an interesting meta-analysis, and I only put it up because of how complete it is. They talk about 159 published trials and 29 meta-analyses of therapies that they looked at, and they basically found three things that had an effect: prone position, neuromuscular blockade, and conservative fluid therapy. So we all know about the neuromuscular blockade story, and we all know about the fluid restriction study. So if you consider neuromuscular blockers and Lasix to be direct therapies, then that's all we have. We don't have a direct therapy. This is an old quote, but it's still true about researchers. We do tend to take failure lightly and just keep moving on. And this was said by Winston Churchill, and this is true today as it was then. So here I am with my list of emerging pharmacologic therapies for ARDS. So aspirin. Aspirin's interesting because we know a lot about the biology of aspirin, how it works. It's cheap. We understand the side effect profile pretty well. And if we could, it might be a good drug as a preventer. There's lots of data in preclinical ARDS models to suggest that it might help. And there's sort of clinical associative data suggesting that patients who come in on aspirin into the ICU are less likely to develop ARDS. This is just one of these studies from the Mayo Clinic, Ogi Gajich's group. And they show that if you were on any antiplatelet in this particular study, you were less likely to get ARDS. And there's a major study ongoing at the moment, which is the LIPS-A study. This has been done in the US, and they're looking at aspirin as a preventer. So it's a phase 2 randomized trial, 400 adults admitted to hospital with a high risk for ARDS. And this risk is judged using the LIPS score, which is a score devised by the Mayo Clinic group. And the intervention is simple. It's a high-dose aspirin for 1 day, followed by a cardiovascular dose up to day 7. And the primary outcome is development of ARDS. Now, this study, recruitment is completed, analysis is in progress, and I imagine we'll have some news by Berlin on that one. Nebulized heparin is another interesting contender. The rationale here is that activation of coagulation is important in the pathogenesis of ARDS, and we all know the hyaline membranes of ARDS and the fact that these are basically fibrin clots. We have good preclinical data to suggest, again, that heparin, when inhaled or nebulized, may be beneficial. And we have a phase 1 study. This is a study of 50 patients done in Australia and New Zealand, looking at nebulized heparin, 25,000 units, either 4 or 6 hourly, versus placebo. And what you have here is ventilator-free days, or the percentage of patients ventilated over time. And what you see is that the patients that got the nebulized heparin came off the ventilator faster. And there is a phase 3 now, there's a phase 2 now in progress, 250 patients with a PTF less than 300 who are just 24 hours or less on a ventilator. And it's nebulized heparin, 25,000 units every 6 hours. So again, this study is planned. You can actually look it up at clinicaltrials.gov, but it's not yet commenced recruitment. Angiotensin converting enzyme is an interesting molecule. We all know that this is highly expressed in the lung, and it cleaves angiotensin 1 to angiotensin 2, and we know that angiotensin 2 is a vasoconstrictor and pro-inflammatory agent. We also know that ACE2 is a homologue of ACE, and this actually cleaves Ang2 in a different way to make a molecule called Ang17. And this blocks many of the effects, are the angiotensin 1 mediated effects. The preclinical studies are, again, reasonably convincing. Imai and her colleagues in Vienna found that ACE, Ang2, and AT1 receptor function as sort of lung injury promoting factors, whereas ACE2 seem to be protective. And this is a nice preclinical study from Giacomo Bellini's group and Antonio Pezzenti, where they looked at Ang-17, they infused it into rats that were subject to an injury of acid aspiration and ventilation-induced lung injury. And these are the animals that got the control, and these are the animals that got the Ang-17 infusion. And so you see much reduced injury, and very interestingly, they also got less fibrosis later on in the pathogenesis. So there is a trial of recombinant ACE2, it's a pharma trial, and they're looking at patients within 48 hours. It's an early phase 1, phase 2. So the phase 1 is a 5-patient study for tolerability, and then there's a phase 2A 60-patient study, and this study is currently recruiting. Adrenomedulin is another interesting potential therapeutic compound. It's an endogenous 52 amino acid peptide, and it binds to the calcitonin gene receptor and appears to have beneficial effects in terms of maintaining epithelial barrier function. And as we've heard from many speakers, that seems to be a key target in ARDS. It's upregulated when you look for it in patients with sepsis or ARDS. And again, there's good experimental data to suggest that it may be beneficial. This is at a slightly earlier stage. This drug has just been given an orphan drug license by the European Medicines Agency, and so clinical trials are being planned at this point. And this is a preclinical study of adrenomedulin in ventilator-induced lung injury rats. So this is where they're given Evans blue, which is a marker of permeability. This is low tidal volume in normal animals. You can see there's very little Evans blue. High tidal volume ventilation or ventilation-induced lung injury, there's a big permeability leak, and this is prevented by the adrenomedulin. KGF, or keratinocyte growth factor, is a fibroblast growth factor which is thought to have beneficial effects and is expressed by epithelial cells and on macrophages. Again, there's a strong preclinical evidence base to suggest that intratracheal KGF may be beneficial in hyperoxic injury, ventilator-induced lung injury, and pneumonia models. And one of the things that's good about this is a drug that's already in clinical use, so it would be easy to translate if it was found to be beneficial. There's a study from Danny McCauley's group in Belfast where they gave palifermin to volunteers with after inhaled LPS, and what you find is the group that got KGF have increased amounts of surfactant protein D, which suggests that they've got type 2 pneumocyte proliferation, and also they got improvement in the percentage of phagocytic cells in the alveolus. They're now running a trial called the CARE trial, which is looking at palifermin in acute lung injury patients. It's a phase 2 study in 60 patients, and the intervention is an IV infusion of KGF versus placebo for up to 6 days. Again, this is a study that recruitment is completed, and so the results should be available soon. Vitamin D is an interesting one in that we know that vitamin D deficiency is really common in critically ill patients. And we also know that vitamin D modulates the immune system. There are preclinical studies to suggest that if you give vitamin D either intravenously or intratracheally, you can modulate the injury and the immune response. And there are some clinical studies as well. So this is a study from Dave Thickett's group in Birmingham, and they looked at vitamin D deficiency in their patients undergoing esophagectomy and patients with ARDS. And what they showed very nicely was that when you looked at the risk of developing a lung injury after esophagectomy, these patients who had low vitamin D, or less than 20 nanomoles, had a much higher risk than those who had a vitamin D over 20 nanomoles. They went on then to actually take rats and make them vitamin D deficient. And what they did then is they gave them an endotoxin injury, and what you find is the animals that are vitamin D deficient, which are in black here, have a much higher lung leak compared to those that are not vitamin D deficient. So another sort of therapy that Dr. Bellingham has already referred to is mesenchymal stromal cells. And these are particularly interesting and a focus of a lot of research. The classic source of these is the bone marrow. And they're actually these cells here in in green. And what they normally do in the bone marrow is they nurture the hemopoietic stem cells. And in fact, when hematologists do bone marrow transplants, they take the mesenchymal cells and throw them away. But what's nice about them, or what's interesting about them, is they have a very low MHC class 1 expression and no MHC class 2. So this raises the potential that you could take them from one person and give them to another patient. So we could have somebody like our friendly Dracula here who's giving blood, where we could take his mesenchymal stromal cells, we could culture them and passage them in vitro, and then we could store them just like we store platelets or FFP, have them on our shelf ready to go for our ARDS patients. And for an acute disease like ARDS, this really is the only way I think we're going to get cell therapies to work. There's good biological data for them, and this is work from Michael Mathey, who, as Jeff said, has really spearheaded the field in terms of translating MSC therapy to clinical practice. And this is one of their earlier studies where they looked at E. coli-induced pneumonia in mice. And what you find is the animals that were given MSCs, which is the broken line here, had a much better survival compared to either the PBS or the fibroblast cell control animals. And when you look at the histology, the MSC animals have much better preservation of lung histology. We've been looking at this for a number of years, and my colleague, Ger Curley, has done a lot of this work. He initially asked the question about repair. So stem cells, the initial concept for them was that they would help to repair lungs. So he devised a model where he caused a ventilation-induced lung injury, then monitored the animals after the injury to look at the repair process. And he gave the MSCs after he induced the injury. And what he saw was, and you can see it here just pictorially, these are the animals that got vehicle therapy, and these are the animals that got MSCs. And what has happened is the lung has restored its function. Now, before you got the MSCs, the lung would have been very heavily neutrophil infiltrated, so it actually cleared and aided the repair process. We went on then to look at this with human MSCs, and this is work done by Claire Masterson and Mairead Hayes. And what they did is they took human MSCs in the same model of ventilator-induced repair and found that the human cells worked just as well. And so human cells restored alveolar PO2, reduced lung leak in terms of BAL protein, and improved the histological recovery compared to either fibroblasts or vehicle therapy. We went on then, this is work from James Devaney, where we looked at E. coli-induced pneumonia. So he showed that human MSCs reduced the injury caused by E. coli pneumonia. And again, this is a micrograph from an E. coli pneumonia animal that got vehicle versus an E. coli pneumonia animal that got MSC therapy. So we went on to try and figure out how it was doing this. And what we found was that the MSCs actually reduced the lung bacterial counts. So it was doing something to help the immune response to eradicate the bacteria from the lung. You can see here, compared to the fibroblast or vehicle, there are less bacteria in the lung of these animals. Ger Curley has taken this work on to try to understand the mechanisms. And this is work he's currently doing in Toronto. And what he has done is he's taken macrophages and cultured them alone or with mesenchymal stromal cells. And what he finds, and then expose them to E. coli. Now, the E. coli, when they fluoresce green, and when they're outside the cell, they fluoresce red. So these are the cells, the macrophages alone. And what you can see is when you look at it under confocal, there's a reasonable number of the bacteria that are green, which means they've been taken up by the macrophage, but there are also red and yellow ones, which means these have not been phagocytosed. When they are co-cultured with MSCs, you can see there's almost complete phagocytosis. And he's looking at the mechanism by how this happens, and he's shown that this is a receptor-mediated phenomenon. I always put this up because it reminds me that we're very good at curing rat ARDS, but not quite so good at human ARDS. But there is nice data to suggest that there is potential in humans. And this again is work from Michael Mathey. And what they did is they took isolated human lungs and that had been rejected for transplant because they were injured in some way. And they then caused an endotoxin injury in these lungs. This is the lung histology from a normal lung. This is the histology from a lung that got LPS injected into one of the lobes. And then they gave MSC therapy in the perfusion pulmonary circulation. And what you see is there's a resolution of the LPS or endotoxin injury. Interestingly, they also found the same thing when they just gave the conditioned medium. suggesting, as Jeff said already, that this is a paracrine effect or something the cells are making. And as Jeff showed, he's gone on to show in a phase 1 and phase 2 study that these cells are safe. There are at least 3 other studies currently in progress. The phase 2 of this study is underway and will report probably next spring. There's a study in the UK, and there's also a study likely to start between Ireland and the UK in the next year. So in terms of emerging therapies for ARDS, we don't exactly have a great track record. As I've said already, it hasn't stopped us continuing to try, and I think we do have to continue to try. There are several promising candidates in early phase trials. ARDS would be really, or aspirin would be really useful if it did help prevent ARDS. I think nebulized heparin is very promising. The vitamin D replacement story is particularly exciting. I think there are a couple of other studies about to report on this, and I think it's a space we need to watch. Statins, I didn't talk about statins, Jeff did, but there may be a HARP3, again going back to the idea of we never stop trying. And that's on the basis that there was a small effect. Drugs for the future, I think interferon is a really exciting drug. And I think maybe a little bit further down the track that mesenchymal stromal cells might also be a possible therapy. But there are likely to be more disappointments ahead. Thank you.